Good evening, dear participants. Welcome to a fresh session of this plant genomic and bioinformatics workshop. Today, we have with us again Ensemble outreach team, and I am handing over session to Dr. Elina Mustar. Over to you, ma'am.
Thank you very much, and welcome back, everyone, to the second day of the Plants Genomics workshop. And just a minute. So my name is Alina Mushtak, and I am part of the Ensemble outreach team with Ben. So Ben presented about the introduction to Ensemble yesterday. We looked at the region in detail, and today we're going to talk a little bit more about the genes and transcripts that you can find in Ensemble. So before I start with my presentation, I'd just like to remind you that we have a training page. I've also shared the link for it on the Zoom chat. And so this is what it looks like. And over here, we have the PDF version of the presentation. So this is a PDF from with the presentation from yesterday, and then my presentation from today. We also have a living document. So this is a shared Google document. And I can quickly show you what it looks like as well. So basically, this document can be used to ask questions. We have a little bit of the course overview, a little introduction to Ensemble, some golden rules for efficient online training, and then we have some space for questions. So this is there because it can serve as like a nice sort of like a knowledge database, because Zoom chat doesn't have threading. So if there are a lot of questions, then I might miss your question. But if you type your question over here, then we can always get back to it. If you are stuck somewhere, for example, when we're doing the exercises, you can always like share screenshots, and then you can also come back to this document later on and refer to other people's questions and our answers to them. So just thought that I'd share this document, and I'm just going to close this for now. And then on this page, you can also find the demonstrations and exercises. So the way that we're going to run this course is that I'm going to speak to you about a little bit of the introduction and the background on genes and transcripts in Ensemble and the data that we have on this. And then we're going to move on to the demonstration. So I'm going to go to the Ensemble Genomes, Ensemble Plants page, and I will show you what you can find, for example, the information that you can find on the Gene tab, and then information that you can find regarding transcripts. And then after the demonstration, you can we will have some time for the exercises. So on this training page, in the Ensemble Plants and Genes and Transcripts tab, you will be able to find this tab for demo. And over here, we have these nice screenshots that are labeled. So if you want, you can follow me along on my screen as I go through the demo, or if you prefer, you can just go through these screenshots and the text here and try these, try to like go through these, follow them on your own machine if you want. And then we have the space for exercises. So there are quite a few of them for this module, but please don't feel like you have to do all of them. This is also not a test. This is just for you to go beyond the demo and try and explore the genes and transcript data for yourself. So, okay, so that was a little bit of the introduction to the structure of the workshop. But now I'm going to move on to my presentation, and we're going to go into the slideshow mode. Are there any questions so far? Okay, there seems to be no questions, so I'm just going to move on with my presentation now. So we're going to talk about genes and transcripts in Ensemble. So Ensemble Genomes does not carry out primary annotation of protein coding gene models. So the gene models can be imported either from annotation in INSDC, sequence archive records, or from other public sources. In addition, Ensemble Genomes is also involved in collaborations from which manual annotation is imported. And then we have manual annotation also to correct errors in automatic annotation. And as Ben also showed you yesterday, for each individual assembly, we have available the information pages from the browser. So you went to the species page, and then we looked at more information about that species and some information and statistics on their gene annotation data that we have. And this page describes in outline how this assembly was created, and it provides a stable third-party identifiers for the assembly. So on my slide, I have examples of sources for the gene annotation for different species in Ensemble Genomes. And here I'm showing you the region in detail view that we've also explored in the demonstrations yesterday, and you might have also come across it during the exercises. So on this slide, we have different transcripts of a gene. So again, a gene in Ensemble is a collection of transcripts, and it's absolutely normal for many alternative transcripts to be transcribed from a single locus. They're also known as isoforms or splice variants. So all of these alternative transcripts are biologically valid and real, and they could be expressed differently. So they could serve different biological functions. So if we focus on the transcripts on the slide, for example, you'll see that, so these are the transcripts over here, and you'll see that the transcript biotypes are color-coded. So we can distinguish between protein coding genes. So these are genes which produce proteins and are translated, and then we have some non-coding genes which do not result in a protein. So for example, these could be RNA genes. So the protein coding genes can have protein coding transcripts which are translated, and these are shown in red. So we also have a legend on where you will have the genes data track. So for example, over here, it says IWGSC, high confidence protein coding. But protein coding genes might also have non-coding transcripts which do not get translated. So those, for example, can be retained intron transcripts or nonsense mediated decay, and these non-coding transcripts are shown in blue. Then we have exons. So exons are shown as boxes or empty boxes. So empty boxes, empty exons are non-coding. So this means that they're not translated, they're only transcribed. Therefore, transcripts of non-coding genes, for example, are composed of empty boxes. And filled or colored in exons are coding. So you can see that a coding transcript will be composed of these filled boxes. However, coding transcripts might also have empty boxes, so empty exons at either 5' or 3' end. And these are UTRs, so these are untranslated regions. So it's important to realize that not all exons are coding. UTRs are exonic as they remain in the transcript after splicing, but they are non-coding as they don't get translated. And then we have these introns shown as these lines that connect the exons with the boxes. And then we have a few hints on the orientation, on gene orientation, so the strand on which the gene is located. So the first hint would be the relative position of the transcript to the genome assembly. So as you may have learned from yesterday's tutorial, yesterday's workshop, that a forward-stranded gene is shown above the genome assembly, whereas the genome assembly is shown as the blue bar. And then the reverse-stranded genes are shown below the genome assembly. Another hint is the arrows next to the gene names. So for example, if you have the transcript names, then the arrows next to them will be pointing in either forward or reverse direction. And then the last hint is basically the shape of the introns. So introns of forward-stranded genes are pointing upward. So they have this mountain-like structure, whereas introns of a reverse-stranded transcript will be pointing downwards. So they have this valley-like structure. So this V. And here we have some sources of the imported annotation that we have in collaboration with or by the scientific community. As I mentioned before, that the genome assemblies presented in Ensemble genomes, so in Ensemble plants, are provided by a variety of sources. But Ensemble genome follows a policy that is compatible with the browser genome release agreement of only using assemblies that have been submitted to the INSDC archives. And this comprises of ENA, GeneBank, and also DDBJ. So that's the DNA data bank of Japan. The last thing that I want to mention before I move on to the demonstration is a few words about gene ontology. So human language is ambiguous, and there are many synonyms and multiple terms that we could use to describe the same thing. So it's the same in the scientific community. For example, one person could be describing innate immune response, and the other person might be calling it non-specific immunity. So we know that they mean pretty much the same thing. However, computer searches don't cope with this very well. And this is why the scientific community came up with this gene ontology terms, which is basically standardized language, which is easily searchable. It is controlled to talk about gene function to make sure that we're all talking about the same thing. And all of these terms also have accession numbers, they have a name, they have a type, and a definition that you can find on Ensemble pages. And I will show you how you can get this information when I move on to the demonstration. Okay, so now it's time for our demo. So we're going to look at the Arabidopsis thaliana PAD4 gene. This gene encodes for the lipase-like PAD4 protein that is predicted to be involved in defense responses. So we're going to find out more information about this gene and about its transcript. So I'm going to jump out of my presentation now. And I'm going to go to Ensemble Genomes. So this is ensemblegenomes.org. And from here, I'm going to focus on Ensemble plants. So I'm going to click on this green button that says go to Ensemble plants. And this is now going to take me to the Ensemble plants homepage. So you've looked at this homepage yesterday with Ben, but today we're going to go further in and we're going to explore the Gene tab and the Transcript tab. So from this homepage, I'm now going to look for the PAD4 gene in Arabidopsis thaliana. So I'm going to focus on the center search section. And from this all species drop-down menu, I'm going to choose Arabidopsis thaliana. And now I'm going to type in my gene of interest. So this is PAD4. And once I'm happy with my specification, I'm now going to click on Go. So we have our search results here, and this is the gene ID that I am interested in. So it is 83G52430. So this is what I'm going to click on. So this takes me to the Gene tab. So this tab collects information on genes, and here we have the Gene tab that's opened next to the Location tab, which opens automatically to show you where the gene sits in the genome, so you can toggle between these two. So then we have a page-specific menu on the left-hand side, which lists all of the available gene views. And similarly, we also have these green buttons at the below this menu. So you have the option to configure this page, add custom tracks, export data, or share this page, and also bookmark this page. So our main view, if I just scroll back up a bit, is we have this little overview, which gives you gene name, Ensemble stable ID. We have the gene description, the synonyms, the gene location, and the strand. So it's on the forward strand, this gene. And then we also have a transcript table. So you can either hide this transcript table or you can show this transcript table. And within this transcript table, we have the transcript ID. We have the length of the transcript. We have the protein length in amino acids, the biotype, so it's a protein coding transcript. If you hover over this, for example, it will also give you a little description of what protein coding over here would mean. And we have this red color coding next to it as well, as I mentioned before, that protein coding transcripts are shown in the red color. Then we have the UniProt match. We have the RefSeq match over here as well. And then I'm going to focus on this gene-based displays menu on the left-hand side. And from here, I'm going to click on sequence. So we're going to have a look at the sequence of the PAD4 gene in Arabidopsis thaliana. So on the left-hand side, I'm going to click on sequence. I'm also going to just zoom in a little bit, just to make my page a bit more clearer. Okay, so here we have our sequence view. And this view shows you the sequence of our gene of interest. We also have its flanking region. On this page, you also have a page-specific help. So wherever you see this question mark icon, this is going to open a new window that explains basically what you see on this page. Then we have options to download the sequence. So you can choose, I think, from FASTA and RTF. So let me just show you. If I click on download sequence button, we then have the option to choose the file format. So you can choose from FASTA or RTF, which is basically, it is rich text format. It's going to give you a static image, so you can see all the coloration and decoration, and this can be opened with a word processor. Then we have some settings. So you can choose the sequence that you want to export. So you can export genomic sequence, which is selected by default, but you also have the option to export just the exonic sequence or the amino acid sequence, the coding sequence, or even cDNA. You also have the option to choose the flanking size. So we can choose the 5' flanking sequence and also the 3' flanking sequence. And then we have these guides that basically show you what the FASTA format would look like and what the RTF would look like. We're not going to download anything right now, so I'm just going to jump out of this menu, either by just clicking anywhere outside this menu to go to the original page, or we can just click on this, this tick button over here. Okay, now we have our sequence over here, and then the coloration that you see on this also has a legend above the sequence. So the legend that you see over here basically explains that all exons are highlighted in this peach color. On top of that, the exons of our gene of interest have a brownish lettering. So these exons, if you were to see them, not over here, it's not very clear, but for example, if you had some peach highlighting, but the lettering was black, then they would be the neighboring genes which also overlap this region. But for example, over here, we see this peach highlighting and the lettering is brown. So this means that these are exons of our gene of interest, so of PAD4 gene. Okay, the next thing that I want to show you is, as I mentioned before, that you also have the option to blast this entire sequence. But if you want, you can also select a sequence and then only blast the selected sequence. So you'll have to click and drag across the sequence to select it. You also have the option to customize this view. So on the left-hand side, if we click on configure this page, you can manipulate this view. So you can choose the flanking sequence, so the flanking length. So for example, it's set to 600, but if you want, you can change it to 50. And let's do that for the 3' as well. You can choose the number of base pairs per row. You can choose to show variance, for example. So right now it's selected to no, but let's say that yes, we want to view the variance in this sequence. And then you can choose further how you want to filter your variance. So I've changed the flanking sequence size and I've asked to show the variance. So now I'm going to save and close this menu. So you'll notice that our flanking sequence is now shorter and we have all the variants marked up on this sequence. So all of these variants are color coded. So if you click on a variant, for example, I've clicked on this R over here, then it will just give you more information about the classification of the variant, the source of the variant, the alleles associated with it, and the color coding that you see is basically it indicates what type of consequence you might have. And if you want to explore that variant further, you can just click on this variant ID, which is going to take you to the variant tab. But I'm not going to go further into this because we have limited time today. But I just wanted to show you that you do have the option to show variants on your sequence. And then from there, you can further find out more information about your variant of interest. There are some questions on here. And I think Ben's answered most of them, but I will also get back to the questions after the demonstration. So please do keep them coming. Please, you can also leave your questions on the living document. So if we don't get back to them within the time period today, then we can get back to your questions later on, or feel free to email us on help desk. But I'm going to move further on with my demonstration. So the next thing that I want to show you is gene ontology. So on the left-hand side, again, on the left-hand side menu, we have this section that says ontologies. And over here, you can find gene ontology for biological processes, for molecular function, cellular component, but also we have plant ontology for plant structure development, and also plant ontology for plant anatomical entity. So I'm going to click on this gene ontology biological process. Just going to give it some time to load. So here we have our table for biological processes. And basically this table summarizes the different ontology terms that are associated with our PAD4 gene. And we have the gene ontology accession number. We have the evidence, the annotation source for this ontology, the mapped, so further documentation. And then we also have the transcript IDs. So we have transcript IDs here, but not gene IDs. And this is because gene ontology terms are associated with a transcript or proteins. Okay, another thing that I want to show you is the gene expression. So the next view that we're going to explore is on the left-hand side. I'm going to click on gene expression. It's given me an error. Let me just refresh this page and hopefully it will work. Nope. Oh, nice. Okay, so you'll see that this is, so this page basically loads external data from the EBI Resources Expression Atlas, and which determines the baseline and differential gene expression for genes. So you'll see that we have this, this interactive widget over here. So if you hover over it, it basically highlights the different organs of the plant. And then on the right-hand side, we have this heat map that summarizes the gene expression across the different tissue, across different projects. And the coloring basically indicates the level of expression. So stronger color would indicate stronger expression. And it's all explained in the legend below this heat map. And then the last thing on this gene tab that I want to explore with you is the external references. So also on the left-hand side, I'm going to click on external references. And over here, we have the external references. So gene corresponding to the different databases identifiers. We have the table for this here. You'll see we have expression atlas, we have NCBI gene, we have a data from TARE and TARE gene name. Okay, and now we've looked at the gene tab, but we're going to now explore the transcript tab. So I'm going to click on this transcript ID from this transcript table, and this is going to take me to the transcript tab. So I'm choosing this transcript ID. So it's AT3G52430.1. Okay, so this is the transcript tab. So you can find the sequence information on it. So for example, we have some statistics. So this transcript has 2 exons. Both of them are coding. We have the transcript length. We have the translation length as well. Then we have the UniProt identifiers for it, the type of this transcript, and also the annotation method. So the gene annotation source. And what we're going to do is that we're going to focus on this left-hand side menu that says transcript-based displays. And from here, we're also going to explore the different sequences. So in the sequence section, we can explore either exons, cDNA, and also protein sequence. So I'm going to first click on exon sequence. And here, I'm just going to hide this transcript table so that our view is a bit more clearer. So over here, we have this table that summarizes all of the exons in this transcript. So you can find the exon ranking transcript. We have the exon stable ID, the start and end position, start phase and end phase, the length of the exon, and then we have the sequence. So you can see that the sequence is color coded. So green indicates flanking sequence. Then we have UTRs in orange. And then we have the translated sequence shown in this blue lettering. And you'll notice that the introns are shown in gray. All of this is explained in the legend above as well. And then you have the option to download this sequence. So so let's click on this download sequence. And then from here, you can select what you want, what kind of format you want first. So it's from FASTA or rich text format. And then you have available settings that you want to include. So you can choose from these various options. And then again, a guide to show you what the different formats would look like. Similarly, I'm not going to download anything for now. Similarly to the sequence page that I showed you on the Gene tab, we can also configure this view that you see. So on the left-hand side, we're going to click on this green button. And you'll see we have this different display options. So we have we can choose the flanking sequence size at either end of the transcript. We can choose to show the full intronic sequence, as that's not shown by default. We can choose to show exons only, add line numbering, show variants, and then also we can hide or filter variants depending on your preferences. So this is what the exonic sequence would look like. And now we're going to go back to this sequence section, and we're going to explore the cDNA sequence. So that is the sequence of a mature spliced transcript. So this view is composed of 3 rows. So the first row is the actual cDNA. So you'll notice that the UTRs are indicated by this yellow highlighting. And then I'll just scroll further down. And then you'll notice we have in the second row, we have the coding sequence. So it's the CTS, and it starts from ATG. And then the third row is the protein sequence, and it starts with an M. So it starts with a methionine. Then we have all of this, by the way, is also mentioned in this legend. You'll also notice that we have for exons, we have black and blue lettering to basically just indicate where one exon finishes and the next starts. And then you can find all of this information by clicking on this question mark icon, which will, so I'll just show you this help page. If I click on this, so basically it's going to give you information of what you can see in the first line and the second line and the third line of the sequence, and also gives you information on the sequence coloring. And similarly to other sequences that we've seen previously, you have the option to download this sequence, and you can also blast this sequence. Okay, so the next thing that I want to explore with you is the protein information. So proteins are not features in Ensemble, so they don't have a dedicated tab, as they do not map to the genome directly. They map to the genome via transcript objects. And you can find a bit of protein information on the Transcript tab. And this includes, so over here, you'll notice on the left-hand side, we have this section that says protein information. So we have protein summary, we have domains and features, variants, if available, the PDB 3D protein model, and the most exciting AlphaFold predicted model as well. So to start with, let's look at this protein summary. So this is basically going to show you a graphical representation of this translation. So here we have the N terminus of the translation of this protein, and then we have the C terminus of this translation. And you'll see that it is colored to show you the exon boundaries. So you can see that we have this pink in color first, so it's the first exon, and then we have the purple color indicating the second exon. And then we also have some protein domains predicted by different methods, and you can find all of this information also summarized in a table format if you like tables better than graphical representations. Then if I go to domains and features on the left-hand side under protein information, you can here find a list of predicted protein domains in a table. So you can find the domain source, the start and end description, the accession number, and the hyperlinks to external sources. All right. And then we have this AlphaFold predicted model. So on the left-hand side, again, under protein information, I'm going to click on AlphaFold predicted model. And these will show you the structural predictions for the PAD4 gene. I'm just going to zoom out a bit so that's a bit more clear. So here we have our interactive molecular viewer that visualizes the structure. So you can rotate this. It is quite interactive. You can play around with it. You can also basically select to zoom in further by clicking on these different letters. And then we also have, this page also displays the data from the 1001 Genomes project that highlights the position of the deleterious variants on this 3D structure. And then you can just reconfigure it. So you can reset the camera. And then you can also have various different things that you want to play around with. You can also screenshot it, download the screenshot, open it in a new tab, and so on. And the last thing that I want to show you is external references. So similarly, like the Gene tab, we also have general identifiers for this transcript. And then another link that we have for external references is the oligoprobes. So let me just click on this. So basically over here, this page is going to show you the transcripts corresponding to the different database identifiers that you can use for microarray experiments. Right, so this is everything that I wanted to show you for the genes and transcripts. I see there are a lot of questions in the chat, and Ben has replied to quite a lot. So whilst we're doing the exercises, I'll try to get back to some of the questions as well. We're now going to do the exercises for genes and transcript module. And as I mentioned, this is on the, so just scroll back up, sorry. So this is Ensemble plants, genes and transcripts on the training page. And if you expand the Ensemble plants, genes and transcript exercises, you can find the exercises over here. And let me just also share this training page. Okay, so we will give about rest of the rest of the time for you to explore the exercises a bit, but I will come back in about 10 minutes time to basically just say some closing remarks, and then the floor will be open for questions. So there is a question about finding homologs for a gene in different organisms. So for example, over here, I've chosen wheat, and I'm on the gene tab, just using an example gene here. And from this gene tab, you can, if we focus on this left-hand side menu and the gene-based displays, you'll see over here that we have this section that says Plant Comparer. And over here, you can find the genomic alignments, the gene trees, the gene gain-loss tree, and then we also have orthologs and paralogs. So I'm going to click on these orthologs. And you'll see that we can find the summary of orthologs of this gene over here. So you can basically choose to display the orthologs for one or more groups of species from this table. And then this table, the table below, will basically give you the individual orthologs in the different species. So I hope that this answers your question. So there's a question on the chat regarding searching for multiple gene names at the same time. Yes, you definitely can. And there are a few options. So you can use Biomart. So this tool is also available over here on the green navigation bar at the top of the page. So this is basically an online data export tool that you can use, and I can show you quickly what it looks like as well. So from here, for example, you'll have to choose the database. So let's say we're interested in plant genes, and then you can choose your species of interest from this drop-down menu. So let's say Arabidopsis thaliana. And then from filters on the left-hand side, you can then, if we select this gene tab, you can then input your gene IDs over here. And then after you've input your gene IDs, you can go to attributes. So attributes are things that you want to appear in your output. So then you can select the different information on genes that you might want. We also have some external information like gene ontology, plant ontology terms, and also external references. So this is one way, but then another way to get information on multiple genes at the same time is via REST API. So that's our language agnostic option, and basically helps you get our data programmatically. And I will speak a little bit about it when I start with my closing remarks. And I also wanted to ask about how everyone is getting on with the exercises. If there are any questions regarding the exercises, please do let me know. Okay, so we still have incoming questions, but I also wanted to bring your attention to our feedback survey. So we would really appreciate your feedback, as it would help us improve our courses, and you can also get a certificate after you fill the survey out. So I'm going to share the link with you. This feedback survey is also available on the training page, but I'll share the link with you so you can access it directly. And as I mentioned, that the feedback that you give us here will help us improve and make our courses better for the future participants, or if you were to attend in the future as well. And you can get a certificate of attendance after you fill the survey out. You can fill it by tomorrow, but it won't take you more than 3 to 5 minutes. It's a super short survey. And there's a question about the gene IDs that you need to give in Biomart. So that's completely up to you, what kind of data that you have on the genes. So if I just go back to filters where you can provide your gene IDs, and so I have selected input external reference ID list, and we have all of the possible gene IDs or the transcript IDs or identifiers that you can choose from in this drop-down menu. So you can choose from gene stable ID, which are ensemble stable IDs, but if you want, you can also provide gene name, gene synonym, NCBI gene IDs, and so on. Hi, Poonam. As Ben has mentioned before, that you can't really perform a gene ontology enrichment analysis or a pathway analysis for a gene or for a set of genes that you've got, but you do have the option to search Ensemble for a phenotype, for example, or a gene ontology term that you already know of, and then it will give you a list of genes or transcripts or variants that are associated with that phenotype or that gene ontology term. Okay, I hope that everyone is working through the exercises, but whilst you're doing that, and I hope that everyone has filled out the feedback survey. As I mentioned, it is a super short survey, 3 to 5 minutes, not even 5 minutes, not going to take you too long at all. But I will continue with some closing remarks. So Ensemble is a genome browser that integrates 4 types of data. So we've spoken about yesterday and today mostly on gene annotation. And I showed you a little bit of how you can get some of the variants to show up on your sequences, but we do have a lot more variation data in Ensemble. We also have comparative genomics, and we also have regulation data for mouse and for human if you are working with those. You can access the Ensemble data, as we've shown you, by the website. So this includes the main website. So this is Ensemble Genomes. And then we also have our sister site. So that's the Ensemble.org. And then if you are working, for example, with the previous Human Genome Assembly, we also have a site dedicated to GRCH37, which is the previous human genome. You can access our data also by using Biomart, and also by programmatically using our APIs and MySQL. And finally, you also have the option to download our whole genome files using our FTP site. And I have the link for this here on my slide. If you have enjoyed our training, please do recommend us to your friends. So apart from the short course that we've had yesterday and today, we also do a longer course. It can be based on plants or a species of your choice. So we would discuss then about the gene annotation, the variation data that we have, the comparative data, and also regulation if it's on human or mouse. The most important thing that I wanted to mention is where to find help on Ensemble. So we have a lot of help and documentation available. So you can also find longer courses and short tutorials and animations on the EBI pages and also YouTube. If you have any questions regarding Ensemble or any issues or recommendations, we would encourage you to drop us an email at helpdesk@ensemble.org. You can also join our public mailing lists to stay in touch and stay updated. We also have our social media pages, so we're very active on Twitter, Facebook. We also have our blog that we post on about updates and new releases. Most importantly, Ensemble is completely free to use, but we would really appreciate your citations, and as this will help us stay informed about how it uses our resources and how. And finally, I would like to thank the entire Ensemble team and the sponsors that make this work possible. And for you, for staying with us until the end of the course and for your questions. We will stay here until 4 o'clock UK time. So if there are any more questions, please do let us know.
So in the meantime, we would like to thank Ensemble Outreach team, Dr. Ben and Dr. Elena, for accepting our request to be part of this plant genomics and bioinformatics workshop. Ensemble team participation in this workshop magnified our cause of affordable bioinformatics education in best possible way. We are looking forward for our next interaction with the Ensemble team as soon as possible for a joint event. Wishing all the best to the Ensemble team for your future endeavors. Thanks Ensemble team for part of this workshop, and we would like to invite you in our future events also. So in the meantime, if participants have some queries, they can ask. We have a few more minutes, possible, probably 12 minutes more. Also, we'll share the video recording along with the chat history with our participants as soon as possible. You can also find the recording of these two sessions on our YouTube channel. That is Decode Life Bioinformatics Training Institute.
Perfect. Thank you very much for having us and for hosting the workshop. I hope that the rest of this this course goes well, and
Yeah.
we'll work with you soon.
The course is absolutely perfect.
Good. Thank you
Thank
very
you
much.
very much.
And we are happy that your team is a part of this workshop.
Thank you.
We have last 5 minutes. If you have any question, you can raise. Otherwise, we will close the session at 8:30 p.m. Indian Standard Time. So we are in the last minute of this session. We'll close the session at 8:30 p.m. So thanks Ensemble team for your time and support. Best wishes, and thanks our participant for listening wonderfully. Tomorrow we have another session on whole genome bisulfite sequencing. So you can check the workshop schedule on WhatsApp group, and I will share the video recording of this session with you as soon as possible. So thanks Benjamin, and thanks Elena for your sessions.
Thank you very much. Goodbye.
Goodbye.
Thank you. Goodbye.